Good morning and good afternoon, everyone. Today I'm going to give you an introduction of the Kinnex single-cell RNA kit. Long reads are the only known way to see isoforms in single-cell RNA sequencing. This is because short reads are limited to sequencing the ends of the molecule, where the single-cell barcodes and UMI reside. Short reads typically sequence between 75 to 150 base, which means you would only be getting about 50 bases that cover the actual transcript. As a result, differentiating isoforms of the same gene based on only this much information is usually not possible. Hence, short-read single-cell RNA-seq are based on gene expression rather than isoform expression. In contrast, long reads can cover the entirety of the transcript, along with the barcodes and UMI information. Length and accuracy are two critical components for single-cell RNA-seq analysis. Where long read lengths cover the entire transcript, accuracy is required for unambiguous identification of cell barcodes and UMIs, which are often only 10 to 12 base long and highly similar to each other. One base error can result in misassignment of cell barcodes. HiFi sequencing offers both long read lengths and accuracy, making it ideal for single-cell RNA sequencing. To give you an example of single-cell isoform sequencing in action using HiFi, researchers used PacBio to sequence colorectal cancer samples with single-cell. Not only did they detect novel isoforms, RNA editing events, they also detected differentially expressed isoforms in different epithelial lineages. Because they can sequence the full-length isoform, they can also do open read event prediction and match them with mass spectrometry data. By comparing isoforms found using PacBio against existing known transcripts in healthy control samples, they were able to identify 24 neoepitopes that could become cancer vaccine candidates. The author said, "We show that full-length transcriptome in tumor cells with mass spectrometry data from matched patient samples can provide a comprehensive source to detect aberrant mRNA-derived antigens." However, the study I showed you earlier was done without using MAS-Seq or Kinnex kits. Up until then, single-cell iso-seq studies have been limited in its application due to throughput on a single SMRT cell. A new method called the MAS-Seq was invented, where it could be utilized to characterize an entire single-cell library with just a single SMRT cell. MAS-Seq is a general amplicon concatenation method, where short cDNA amplicons, such as those produced by the 10x Chromium single-cell platform, can be concatenated to create larger fragments to be sequenced with HiFi. Because of HiFi's accuracy, this concatenation increases the effective molecular yield without affecting the ability to call single-cell barcodes or UMIs. The MAS-Seq authors demonstrated the ability to obtain 33 million cDNA molecules on a single SMRT cell. In fact, this is in Sequel 2, in a 6,000 cell CDA plus T cell library. Not only was the long-read data able to recapitulate the short-read based cell types, it also revealed isoform information with cell type specificity. Just to give you an example here, they showed that different isoforms of CD45 are expressed in different cell types. The Kinnex single-cell RNA kit is the latest offering from the MAS-Seq kit. It is an upgrade of the MAS-Seq for 10x single-cell 3' kit that offers both sequencing library construction for 10x Chromium single-cell 3' and 5' kit. It takes as input 15 to 75 nanograms of 10x cDNA with a target single-cell library of 3,000 to 10,000 cells. It is a 2-day library preparation process from 10x cDNA to sequencing-ready library. And a new feature from the MAS-Seq kit is it supports barcoded Kinnex adapters. With SmartLink 13.1 and up, the Kinnex single-cell RNA run design is able to generate gene and isoform count matrices that are compatible with tertiary analyses such as SURAT and SCOMPy. On a single Revio SMRT cell, the throughput is going to be 80 to 100 million cDNA molecules. No short reads needed. We've released publicly available Kinnex single-cell datasets. For example, we showed on the matching HG002 Kinnex compared with short-read matching data, we showed high concordance in cell barcodes and UMI abundances. We also show that with Kinnex single-cell, you can get the full-length isoform information. Here we're showing the read lengths going up to 4 to 5 kilobases, and that there are different categories, FSM, ISM, and IC, which are based on the SCONTY3/Pigeon isoform classification system. You can browse this dataset by using the QR code below or going to pacb.com/dataset. If you're interested in knowing more about the Kinnex single-cell RNA kit, here are the resources. You can read the application note. You can see how we compare with other long-read sequencing technologies, or to learn more about general offerings of long reads for RNA sequencing, visit pacb.com/RNA-seq. Thank you for listening to my talk.
So a warm welcome to the webinar today, having the title Understanding Full-Length Single-Cell RNA-seq of Immune Cells in Cardiovascular Diseases. My name is Anastasiya Grinko. I'm a PhD candidate in the Helmholtz Institute for RNA-Based Infection Research in Würzburg in the laboratory of Professor Dr. Antoine Emmanuel Saliba. And today I'm going to focus on the technical aspects of MAS-Iso sequencing, and I'm going to bring you a bit closer to my research project. But before we dive deeper into the topic, I want to give a brief overview about myself and why am I talking about this topic. So as I told you before, I'm a PhD student in the laboratory of Professor Dr. Antoine Emmanuel, and I started my PhD last year in September, where I focus on the analysis of long-read sequencing data. And I mostly focus on developing new types of protocols to explore the dynamics of splicing in the context of immunity and infection on the single cell level. And before we talk about isoforms in more detail, we should ask ourselves, why do we use MAS-Iso sequencing, or in my case, single-cell MAS-Iso sequencing and the Kinnex kits? So the state of art of sequencing as of right now is still based on short-read sequencing. But these short reads do not really allow us to have the needed resolution for isoforms. I always compare them to puzzle pieces, and our short reads are very small puzzle pieces. And these pieces are actually way too small to span the full length of isoform. That way, we cannot really assemble isoforms the way that we want to, because we have lower read coverage. With long reads, we have a high read coverage, which then in turn allow us to have a very good isoform elucidation. And to achieve that, we use Pacific Biosciences MAS-Seq, and thanks to the circular consensus sequencing, we can get very long reads ranging up to 15 kilobases with a very high accuracy of up to 99.9% with the so-called HiFi high-fidelity reads. And to overcome the conventional caveat of the low throughput coming with long-read sequencing techniques, we use MAS-Iso sequencing, as explained in the publication of Al-Khafaji et al. In Nature 2023, which is based on the concatenation of cDNA into big mass arrays. And these big mass arrays, in turn, allow us to boost the throughput significantly after long-read sequencing. So, how does our lab use single-cell MAS-Iso sequencing? We applied single-cell MAS-Iso sequencing to cardiac immune cells in mice challenged with myocardial infarction, and then we obtained the single-cell transcriptomics before and 5 days after cardiac infarction. Or, more precisely, we already had short-read sequencing data from our collaborators available, as you can see here in the publication of Reitz et al. At Cardiovascular Research 2022, And then we just applied long-read sequencing with MAS-Seq or single-cell MAS-Seq onto the cDNA that we already had available. And in the following, I want to present you my data with a heavy focus on the quality metrics, because I believe it's very beneficial for you, as it might guide you to your own research when using Kinnex kits or when using single-cell MAS-Seq or MAS-Seq in general. So first of all, if we look at the quality metrics of the zero-mode waveguides, we can see that actually a lot of the zero-mode waveguides do not pass the filters. I was a bit shocked in the beginning when seeing this, but that's actually completely normal. So if you have something in the range of what I'm showing you right now, you can be reassured this is completely okay and you can continue with your research. More importantly, we can look at the quality metrics of the read quality. So after my sequencing run, we get around 1.7 million big mass arrays with a mean read length of 14,405 base pairs and a mean Phred score of Q31. After the read segmentation of the mass arrays with SCARA, we get around 2060 million total segmented reads or S reads, and they have a mean read length of around 900 base pairs. And the cool thing is also that we have a full array percentage of 87% with a mean array size of 16, meaning that the vast majority of our data is actually in full arrays and allows us to have really good data. And after looking at my data, after checking in, I can say that, okay, this is good data. I can continue with my research. I can continue with my downstream analysis, and I go on with the processing of the data with the conventional iso-seq free and pigeon pipeline. And so even after processing with, for instance, Lima, tag, refine, and correct, so the typical iso-seq free pipeline, where we cut off adapters, where we orientate our reads in 5' and 3' direction, where we tag our reads, where we make sure that we have non-chimeric reads, and where we only retain reads with poly A, we still in the end do not lose a lot through the processing step. And so in the end, I had around 25.5 million reads left with which I can work. And as we had also the complementary short reads, we actually wanted to compare the short reads to the long reads, especially in terms of how many cells do we detect, how many genes do we detect, and UMIs and isoforms do we detect. So what comes pretty clear already in the beginning is that with the long reads, we detect less cells. But this is mostly, or most probably, due to the sequencing depth that is just smaller with long-read sequencing than with short-read sequencing. We also detect significantly less UMIs and genes per cell, but what is good, so most of the genes that we detect are actually mostly overlapping. So the genes that we detect only with one of the sequencing platforms are mostly very lowly expressed genes, and I can show that with my data, but this has also already been showed in other data sets, such as in publications from Mortazavi et al. So at the bottom, you can also see the gene and isoform distribution when it comes to the features that we detect, when it comes to the counts that we detect, and when it comes to the percentage of mitochondrial reads. So everything is very good distributed, and we could continue with our research. So looking at this and seeing that we have a significantly less amount of UMIs per cell and genes per cell, we asked ourselves the question, is MAS-Seq alone sufficient to reconstitute the short-read single-cell analysis? Because optimally, of course, we would like to only have long-read sequencing without complementary short-read sequencing, as this would save us costs and time. And so to be able to see if MAS-Seq is enough to reconstitute our short-read single-cell analysis, we use the so-called adjusted rand index, ARI, which is just a measure of similarity between two data clusterings. And we compare the Illumina short-read data annotation to the PacBio long-read data annotation that we had. And so with an ARI of 0.512, we could actually see that MAS-Seq alone was able to delineate cell types similar to short-read sequencing data. So we were able to just use the long-read data onto our following research. And with that, I subsetted my immune cells for my research focus, which is the elucidation of cell subtype-specific isoform usage with a heavy focus on monocyte and monocyte-derived macrophages, as you can see it here in the UMAP on the left, where we just have different subtypes of monocytes, monocyte-derived macrophages, and resident macrophages. And so here my research questions are there differentially spliced genes between the subtypes? Are there genes that are differentially spliced but not differentially expressed? How are those genes involved in myocardial infarction? And which functional impact does differentially spliced genes actually confer? And here it is very important to stress that before we can actually really dive deep into the underlying biology, there's one crucial aspect that we need to understand. And this is understanding isoforms and classifying isoforms and validating and filtering them. So in long-read sequencing, isoforms are actually oftentimes characterized after their structural categories. And these structural categories are based on the alignment of the isoforms that we find based on a reference transcript. And I'm using the classification of SQuanti-free. I will talk in a few minutes more about SQuanti-free. And the characterization is the following. So we have full splice matches, which are really nice because they match all the splice junctions of the reference transcript completely. Then we have incomplete splice matches, which match the reference splice junction partially. And we have NIC and NNC, standing for novel in catalog and novel non in catalog, which are novel isoforms with either a new combination of known splice sites or a novel isoform with at least one new splicing site. Then we can classify isoforms also as genic introns, meaning that they lay within an intron, and genic genomic, where isoform overlaps introns and exons. But having an FSM is good. We know that this is an isoform, that we can say with almost certainty. But what about incomplete splice matches, novel in catalog and novel non in catalog isoforms? Because especially in the case of incomplete splice matches, how can we be really sure if these isoforms are actually true real shorter transcripts or just either truncated or sequencing artifacts? And with the novel isoforms, so the NIC and NNC isoforms, how can we be sure whether these are true novel isoforms or again, just arising due to sequencing errors? And the answer here is that we need orthogonal validation of isoforms. Because to be really sure if we have true isoforms, we need to confirm them. We need to have some aspects that we can control them with. So we need orthogonal validation. And this orthogonal validation can arise in the following aspects. So of course, we can look at the 5' and 3' sites. For instance, we can use KH-peak data for validating our 5' sites. We can use poly-A sites and poly-A motifs to validate our 3' sites, which is also very important. We can look at the distance to the nearest annotated 5' and 3' sites, which we find in the so-called classification file that I will show to you in a few minutes. Then when it comes to incomplete splice matches, what is a good instance is always the open reading frame prediction, because if we have an incomplete splicing match that is actually protein coding and has an open reading frame, it is more likely that this incomplete splicing match is actually a true short isoform than just a sequencing artifact. Also, one very important aspect that we can look at is the counts, because the more counts an isoform has, the more confidence we can give to the isoform of being a true isoform and not just in sequencing error. And if we have, like in my case, for instance, the complementary short read data, we can also use the short read data as some kind of validation because we can look at the junction coverage. So for instance, when we do star mapping, we can just use the sj.out.tab file to really get the validation of our splice junctions on the short read level. And for me personally, to achieve really true characterization, validation of isoforms, I use Squanti, or in my case, Squanti-free, because Squanti-free is a toolkit for the detailed characterization of isoforms. And what Squanti-free does is that it combines long-read data with reference annotation and orthogonal data, as you can see here, and it consists of 3 different scripts. So we have the isoform classification and quality control with squantiqc.pi. Then we go to the very, very important step of filtering to remove potential false positives from their transcriptomes with squantifilter.pi. And in the end, we can also run squantirescue.pi to rescue the discarded transcripts being false negatives, and keep transcriptome diversity. And just to not go into too much confusion, Pigeon is actually a Squanti-free implementation for iso-seq-free with Pigeon Classify, which is similar to Squanti quality control.py, Pigeon Filter, which is similar to squantifilter.py, and then we have Pigeon Report, and especially for the people wanting to work with downstream Surat applications, we have Pigeon Make Surat, which allows us to make output very compatible to Surat. And so I started with Squanti-free quality control, and for Squanti-free quality control, which is really nice, is that we just need a collapsed GFF from the iso-seq collapse step. And this is our long-read defined transcriptome. Then we add reference data, so the genome FASTA and the annotation GTF, and we also add this extra data, which is just orthogonal validation. And for instance, if you're working with very well annotated organisms such as human and mice, it is pretty easy to find that extra data. And some of the extra data, like some cage peaks or poly-A motifs, is actually also found in the Squanti-free folder if you work with human or mice. And so we add everything to Squanti-free quality control, and what we get out in the end is, first of all, a big Squanti quality control report, which is like this big HTML or PDF file with a lot of plots that show you the quality metrics. It is very helpful to look at that. And then we get the core of the Squanti-free quality control, which is the classification file. And the classification file is just a big data frame with detailed information about each detected isoform after sequencing. And then we also get the junction file, which is similar to the classification file, but now being a very big data frame with detailed information about each detected splicing junction. And just down here, you can see an example of a classification file. So this is what I get, for instance, if I run it on my data. You can see some of the columns of the data frame. So we can see the isoform, we can see where is the isoform found on which chromosome, is it on the negative or the positive strand, how long is the isoform, how many accents do we have, which structural category do we find. We can get the associated gene and the associated transcript to the isoform. We get the reference length and the reference exons, and very importantly, things like the difference to the transcriptional start site, or the difference to the transcriptional termination site, and many, many more. So when I use Squanti quality control and Squanti filtering my data, this is what I get. So I classified my isoforms according to the Squanti categories and subcategories, and then I filtered with the Squanti filter with default parameters. And it is really interesting to see, because before filtering, we have way more isoforms than after filtering. And we can see that before filtering, a lot of our isoforms are actually marked as intergenic, as we can see here, meaning that they most probably are sequencing artifacts. After filtering, we get way less isoforms, and also the proportion shifts. Now, after filtering, our structural categories are more full splice matches and especially incomplete splice matches. And actually, I wouldn't say it is a rule of thumb, but this is something you can really expect after running Squanti quality control and Squanti filtering, is that around 30% of all of your isoforms that you detect are actually incomplete splice matches. And the cool thing with Squanti filtering is that not only do you filter out the isoforms that are most likely just artifacts, you also get the filtering reasons, so why the isoforms has been filtered out. And so, for instance, the biggest reason for isoforms to be filtered out is that they have a high percentage of adenine downstream the transcriptional termination site, meaning that we have some intrapriming events. This is followed by the low minimum coverage, meaning that, especially when we have the short read data accompanying our long read data, that our splice junction have a very low coverage of counts, followed by autocanonical, meaning that none of our splicing sites are actually canonical splicing sites. And of course, and this should always also be mentioned, we can filter out isoform based on potential reverse transcriptase template switching artifacts. And how important filtering is becomes clear when we look at this plot, because actually most of the isoforms that we detect are not true isoforms. Only a small part of all isoforms that we detect are actually true isoforms, because most of the isoforms are just sequencing artifact, with intrapriming events being the most highly abundant isoforms or isoforms that we can find. Nevertheless, if we look at the distribution of counts, we can still see that the true isoforms actually still get the most counts. So we still have around 60 or over 60% of counts that really map to true isoforms. And if we look at the distribution of the read counts, and if we look at the distribution of the length in the different Squanti categories, we see more trends. So for instance, it becomes clear that in full splice matches, full splice matches tend to have more counts and also tend to be longer, whereas things or isoforms that we would more see as an artifact, so for instance the genic or intergenic categories, tend to have less counts and also be shorter. Nevertheless, we should keep in mind here that, for instance, if we look at the novel not in catalog or novel in catalog isoforms, they also tend to have less counts. So here, for instance, we need to be very careful, very cautious, and really find the best ways for us to evaluate our isoforms. So after I classified and I filtered, I was able to get all of my differentially spliced genes. And I was focusing on 3 different comparisons. So for me, I wanted to look at the differential splicing between TREM2 high macrophages versus resident macrophages, TREM2 high macrophages versus monocytes, and TREM2 high macrophages versus MHC2 LB1 macrophages. And not only could I find that we actually do see differentially spliced genes in between the macrophage or monocyte-derived macrophage subtypes, we also see that most of the genes that are differentially spliced are actually only differentially spliced, but not differentially expressed. So what we can see here is that without looking at the splicing, we would lose a whole level of information, because there are changes that we would not see on the gene level. And MAS isosequencing, or in my case single-cell MAS isosequencing, really allows us to uncover that new level, looking at differential splicing rather than just differential expression. And one example that I want to highlight out of my research, one example gene, is FCGR2B, which is a gene that is involved in multiple function in the immune response. And so what we can see here in FCGR2B is that there are 2 very interesting isoforms. We have the isoform 4 and the isoform 68, which are differentially used between the subtypes of the monocyte-derived macrophages, or monocytes in this case. So for instance, the isoform 4 is found more in the TREM2 high macrophages, whereas the isoform 68 is found more in the monocytes. And interestingly enough, both of those isoforms are also kind of distinct to each other, because we can see that in the TREM2 high macrophages, not only do we have an alternative 5' start site, we also have 1 exon less than the isoform in the monocytes. And if I get this data, I look at a lot of criteria for validation, because I want to be sure that those isoforms that I found are true isoforms, and again, not sequencing artifacts. So out of the classification file, as you can see here on the right, I can get multiple validation criteria. So I look at the amount of exons, I look at the difference to the transcription start site, I look at the difference to the transcription termination site, I look at my minimum coverage, my full length counts that I find, and of course if it's an intra-priming artifact or not. And then I can validate that these 2 are true isoforms. And interestingly enough here, we can see that the open reading frame length of both of the isoforms also differs quite significantly. And if the open reading frame length differs so significantly, that might also imply new proteins or different protein structures. And those in turn might even have functional impacts that are very specific to either the TREM2 high macrophages or the monocytes. Of course, for the future, the question remains which functional impact can occur here, and we need to cross-check that. But already with single-cell MAS isoseq, we can actually see that there are differences that we find in between the macrophage subgroups. So very briefly also, what I want to present today is that I do not only want to look at the differences of splicing between clusters, but I was also interested in the changes of isoforms in a dynamic way, so lineage-based splicing. And this is actually very much in their baby shoes. I just started with the analysis of the data, But for that, what I did is that I inferred pseudotime and entropy from my monocyte and monocyte-derived macrophages. So you can see in the UMAP on the right which monocyte and monocyte-derived macrophage subtypes I was looking at. And then I inferred a pseudotime. So I ordered the cells from 0 to 1 on the trajectory to see how well they differentiate. And I looked at the entropy, meaning how likely a cell is to still differentiate into a new status. I looked at the diffusion component, as you can see. So looking at the trajectory or differentiation trends. And then I wanted to know, based on my trajectories, how do isoforms switch or how do isoforms behave in a dynamic way? So based on prior biological knowledge, I set some initial states. So I set my monocytes as my initial state, and I set some terminal states. And with those interplay of initial states and terminal states, I defined my lineages or my trajectories, and I defined and what exactly I want to look at. And so after doing a bit of research over here, we had some preliminary findings. And I want to show you one of the example of the preliminary findings. And this is the gene FCGR2B again. And again, we can find the isoform 4, which is more expressed in the TREM2 high macrophages in our differential splicing test, and the isoform 68, which is expressed more in monocytes after our differential splicing test. And interestingly enough, what we can see here for that gene is that not only do the differential splicing differ between the monocytes and the TREM2 high macrophages, we can also see that the expression of those two different isoforms differs extremely over the time in the distinct lineages. So we can show that not only can isoforms be expressed differentially between clusters, isoforms can also be highly dynamic, and their dynamics should be taken into account. However, as I said before, this data yet is very recent, it is not explored much, but I'm looking forward to looking into this deeper, because I think the dynamics of isoforms and how they are changing over time is not yet explored a lot. So I'm very interested in that. And single-cell MAS-Seq together with the Kinnex kits will allow me to really go deeper into that research. So at the end, I just want to give you a brief summary and recap about the things that we talked about. So single-cell MAS-Seq sequencing leverages the throughput of smart sequencing, allowing us to reconstitute short-read cell clustering and cell type identification, even if we have significantly less UMIs and genes per cell. Squanti-free, or pigeon in the case of isoseq-free, allows us to confidentially classify isoforms and filter out sequencing-based artifacts. Just keep in mind how important it actually is to filter the isoforms, because we can see that most of the isoforms are actually based on artifacts. And then when it comes to the biology, we can see that monocytes and monocyte-derived macrophages present cell type-specific isoform usage in the context of myocardial infarction. And even though in its baby shoes and very recent, we can already see some trends of lineage-based differential splicing to uncover dynamics of isoforms. So with that, I'm at the end, and I hope I could guide you a bit through your research with single-cell MAS-Seq sequencing. I hope I could give you some new information. I thank you for your attention, and if you have any questions, please feel free to ask.